Methyltransferases add methyl groups to a DNA. They most commonly add the methyl group to either N6 of adenine or C5 of cytosine. The type 2 restriction enzymes have cognate methyltransferases. They are used in the native context to protect the genome from the action of the restriction enzyme and thus typically have the same specificity as the restriction enzyme. For example, BAMH1 methyltransferase modifies the fourth C in the BAMH1 restriction site. Upon methylation, the activity of the cognate endonuclease is blocked at that site. DAM and DCM methylation frequently block restriction endonucleases. The lab strains of E. coli encode these two methyltransferases, which modify the sequence GATC and CCWGG. When a restriction site overlaps these sites, the enzyme will be blocked. There are some useful strains like JM110 that have these genes knocked out, but these genes also play regulatory roles in the cell and the bacteria aren't healthy. Whether an enzyme can be blocked by DAM or DCM methylation is cataloged by NEB. One of the more common ones is XBA1. If the restriction site is followed by the sequence TC, it will overlap a DAM site and thus be methylated and blocked. Eukaryotes also methylate their DNA extensively for controlling gene expression. CpG methylation involves methylation of C5 positions in rafts in the genome called CpG islands. Restriction sites overlapping CpG sites may be blocked. DNA from cells can be methylated in ways that prevent digestion by restriction enzymes. Though you can't treat a DNA with an enzyme that pops off the methyl groups, none such exists, replication of the DNA will result in methyl group-free versions of the molecule. PCR products are thus unmethylated.